For today, we're going to be starting our discussion about the immune system, and we're going to begin with innate immunity. Generally speaking, immunology is the study of the body's response to infection by microorganisms. When we consider our ability to be able to defend against things from our outside world trying to get inside of us, this is largely dependent on the ability to distinguish between things that are considered to be self or non self. Things that are self are anything that our body naturally produces: our own proteins, our own sugars, amino acids, etc. Anything that's non self is something that would be considered to be foreign. We would not be natively making these things in our own processes. And so when our body would detect one of these things as a non self entity, that's when all of the alarm bells would start to go off, and all of a sudden we can start to mount a response and act accordingly to remove or destroy that non-self agent. And this is especially imperative in the context of microbiology and its impact on human health. The immune response is divided into two main branches. There's the innate immune response, which is more of a generic and non-specific response. We'll talk about that a lot today in terms of what steps actually occur following that initial infection. And then also, as a higher organism, higher vertebrates especially, there's also the adaptive immune response. This is also called the acquired immune response, and it's much more specific, meaning that it can target certain species of organisms in a very effective way rather than just responding more generally. There are a couple of differences to consider between the innate and adaptive immune responses. Innate immunity is going to have a very fast reaction time. It is the first response that activates following infection and primarily involves physical and chemical defenses. Physical meaning actual structural integrity of cellular layers, for example, at our common portals of entry. Or chemical defenses like the production of antimicrobial things inside of our secretions like mucus or tears. Unfortunately, the innate immune response doesn't have memory, so no matter what type of infection you're getting, that innate immune response is always going to react the exact same way. We're going to clarify this more as we go on. The adaptive immune response is something that is a little bit slower to react and can sometimes take up to a whole week to get into full swing. Usually this kicks in if the innate immune response is not enough to get rid of an infection on its own. And at that point, the body has time to identify what type of pathogenic agent is causing the problem. Is it an intracellular or an extracellular infection? And at that point, it can release a very specific set of cells that can go out and do very tailored work to make sure that the infection type is being adequately attacked. In cases where you might have a viral infection, you're not going to want to have a lot of antibody production, right? Because antibodies are going to target stuff that is extracellular in nature and is more suited for a bacterial infection. Or vice versa, you wouldn't want to have a bacterial infection, but have our suite of cells that are used to fight off diseased or compromised cells being activated instead. So that adaptive immune response adapts to whatever happens to be present within our bodies at the time. And more importantly, it has memory. So if you end up with an infection and you become reinfected with the same species down the line, your body can remember that it saw it before, the methods that it chose to implement to eradicate it from the body, and it can get rid of it faster and better than it did the first time that it saw it. When we talk about immunity, that's going to be the ability of a host to resist infection. And for us, that's done by initiating an immune response. That initial activation is done through reaction to antigens or immunogens. So anything that's antigenic or immunogenic is non-self. Those are those things that are, again, not produced within ourselves as hosts. It's also important to distinguish that something foreign to us doesn't always have to be a living foreign entity. It can be things that are non-living as well, like a foreign object or even things like allergens. Our allergies are a misplaced immune response, because a lot of allergens don't actually pose any threat to us. Our body, though, still responds as if they may, and goes all throughout the same efforts and motions that it would if it were fighting off something very pathogenic. Many of these antigens are usually going to be bacterial or viral specific, things that are not produced in either eukaryotic hosts or human hosts. Things like peptidoglycan, lipopolysaccharide and gram-negative cell envelopes, and also things like bacterial flagella, D amino acids that we talked about ages ago. The list goes on and on. As I mentioned, the innate immune response is going to be our first line of defense against new infections. It is present in all multicellular complex organisms, from plants to insects and also many animals. The mechanisms for the innate immune response are, in fact, innate. They are present in us when we emerge from our mothers into the world at birth. So, as a baby, if you get an infection and if your immune system is normal and intact, you're going to be able to mount the innate immune response. What's interesting is that this particular leg of our immune system will activate the exact same way every single time, no matter what the infectious agent is. If it is a bacterial infection, if it's a viral infection, it doesn't matter. The same types of mechanisms are going to be set into motion. And because there is no memory associated with this, what we see is an identical response every single time. So if we were to get infected and reinfected with the same types of pathogens, our innate immune response would always react exactly the same way. The initiation point for the innate immune mechanisms are at the primary routes of entry into the body. So those common portals of entry, once again, that are exposed to the outside environment, our nasal passages, our oral cavity, our reproductive tracts, our eyes, our ears, all of these things are all regularly exposed to the outside world, and as a consequence, any pathogens that happen to also exist within them. One thing that all of these areas have in common with one another is the presence of epithelial cell layers. Epithelial cells create a physical barrier. They line up side by side. They are tightly bonded to one another, and nothing can normally pass through the epithelial layer. You can consider them almost like bricks in a wall. In addition to their physical limitations, they also secrete a lot of factors that are antimicrobial in nature. So if pathogens do happen to reach those areas and try to settle in through that adhesion that we talked about, all of those antimicrobial substances can kill them prior to them being able to get a good foothold. The skin is probably our very first and best defense against the outside world, and it consists of our epidermal and our dermal layers. You can see those shown here. The epidermis is that top section of the skin and can extend further down as well. And the dermis is found below. So tons and tons of layers of epithelia that are going to, again, physically block passage of organisms from the outside world into our body systems. Of course, when this skin layer is breached through a cut or some other kind of problem, especially if that penetrates down to the dermal layer where there are blood vessels, that can allow a direct point of entry into the body through that parenteral route that we've talked about a couple of times. But either way, in the skin, we have a bunch of immune cells that are there just in case that area does get breached and pathogens make their way into the body through these routes. These are called skin-associated lymphoid tissues, and these are cells that are there to facilitate very fast processing and destruction of microbes on site. Langerhans cells are a specialized skin cell type that can phagocytose antigens. Phagocytosis is one of the primary mechanisms used in our immune system where cells will engulf different pathogens and then either destroy them or take what they've found, migrate away, and go and talk to other cells of the immune system to show them what they've found. That second example of the migration and the demonstration of what was found at a site of infection is more linked to our adaptive immune response, but that is what we're seeing in this diagram here. So you can see that there's an antigen present, and there are Langerhans cells present in the skin layer. They detect this antigenic agent, and then they engulf it. You can see that there's the phagocytic event happening here. And some cells, not necessarily Langerhans cells here, but other immune cells at this point would just destroy the pathogens and continue their giant meal. But in this case, Langerhans cells will take off and go and speak to other immune cells that are found inside of our lymphatic system. Helper T cells are an example of a cell that can be communicated with. But again, this process is more highly linked to our adaptive immune response rather than our innate response. We also have a lot of mucous membranes in these areas of the body. If you can imagine, our gastrointestinal, respiratory, or urogenital tracts, and even our eyes are all covered in different kinds of liquids, whether it's mucus or tears. And these things have a couple of different functions in a couple of different contexts as well. The secretion of things like mucus or tears will actually capture bacteria that are trying to nestle in on cellular surfaces. So as mucus gets produced, it will lay on top of that epithelial layer, and it's quite sticky and thick. So pathogens, as they're trying to get down to the host cell surface, will actually get stuck in there. And when we're coughing or sneezing or expelling this mucus in a variety of different ways, We're actually getting rid of a lot of microorganisms in the process and preventing them from being able to attach to our host cells and multiply. On the eye, especially because of not only the moisture levels there and the saltiness of tears, there's also the physical abrasion of the eyelid scraping across the surface of the eye that helps to protect it. One tiny little cell clinging to the surface of our eyeballs are not going to be able to withstand the force and the pressure of the eyelid. The chemical barriers not only extend to specific products that are produced, but also the pH. Places like the skin and the urogenital tract are typically slightly acidic compared to other parts of the body, which will not be favorable for most kinds of microorganisms. We've talked a lot about how pH will affect the physical characteristics of enzymes and can alter them to an extent where they no longer become functional. Many bacteria fall victim to this if they try to colonize the wrong part of the body. There are also a variety of proteinaceous factors that get assembled and released by our immune cells that can go off and destroy the invading pathogens. Defensins or cathelicidins are two examples of these that will interfere with various cellular functions in a similar manner to what we see with antibiotics. They can inhibit things like cell wall formation, they can inhibit protein synthesis. Any number of things can be prevented from happening, which obviously can kill cells. Lysozymes are one really important class of antimicrobial peptides, and these are enzymes that can actually digest many types of bacteria by cleaving their peptidoglycan linkages and causing the dismantling of the cell wall. As I've said 1,000 times, it feels like whenever we compromise the integrity of the cell, whether at the level of the membrane or the cell wall, the cells just can simply not live on. Transferins are another kind of protein that are important in combating bacterial infections, especially because many bacteria require iron to live. We have a lot of iron as human hosts in our blood and in other types of places in our bodies. And by secreting transferins, we can actually grab our own iron and sequester it away. Sequestration simply means that we take it and hold it in such a way that makes it inaccessible to bacteria, so they can't grab it and use it anymore. It's still present in the body, it's still available for us, but the bacteria are not able to use it. Think of it as putting it up into a high cupboard where a small child can't reach it. It's still there, it's still available, but it cannot be used unless you want it to be. Iron is one of the cofactors that's needed for many different metabolic processes for bacteria, and when they are deprived of it, a lot of them can't survive. We've talked a lot about native flora throughout this course, and we'll discuss them again here in the context of the immune system, but these are non-pathogenic bacterial species that have some kind of symbiotic benefit to living in or on us. These cells are not going to appreciate any invasive organisms coming in and getting in on their turf, trying to take their nutrient sources or competing with them for space. So our commensal organisms will actually defend us to a large extent by secreting antimicrobial things that will act and kill invading organisms on our behalf. While we may consider their actions a little bit selfish, at least we do benefit from their greed in wanting to have us all to themselves. Many different responses of the innate immune system help to protect us from those invasive pathogens. We're going to touch briefly on the inflammatory response, the fever response, and the secretion of antimicrobial substances as well. Many of the mechanisms of the immune system, not only the innate but also the adaptive, are highly regulated by cells. These are specialized cell types that have specific jobs in a very large and well-orchestrated event. And we're going to discuss some of those along the way in the next lecture or two. First and foremost, for a little bit of background to set the stage here, there are two types of organs and tissues that are involved in helping us to facilitate our cell-mediated immune responses. We have our primary lymphoid organs. This is where all of our different immune cells arise from. So there are two locations in the body that will produce immune cells. These are our bone marrow and also our thymus. Every other tissue or organ that's associated with the immune system is considered to be a secondary lymphoid organ. This is where our lymphocytes and our other immune cells can encounter and bind antigen. These include things like the lymph node, the spleen, or organ-specific tissues where we see increased immune activity. Places like the mesenteric lymph nodes of the gut, for example, fit well into this description. This is a diagram here just to generally show the lymphatic system and some of its more important components. Here we have the thymus. You can see that this organ is found in the neck chest area, and that is where some of our immune cells are born from. And then the majority are also born out of bone marrow here. So our two primary immune organs are here, and then the rest of all of these things would be considered secondary. We haven't talked much about cellular differentiation, but most cells of the immune system arise from a single progenitor. You can think of a progenitor cell as a stem cell. And these cells at that state don't have a specific job yet. They're just a cell waiting to be pushed into one task or another, and they can commit to that job and they remain in that job forever after they undergo a differentiation. But either way, all immune cells arise from what are called hematopoietic stem cells. And then from there are two branches. We have the myeloid stem cell branch that produces many of our cells that we're going to talk about today. Lots of these are phagocytic, not all, but most are, and they are typically white blood cells. You might recognize a couple of those names from other courses. And then the other branch, of course, is the lymphoid stem cell that will produce our two major types of lymphocytes. The lymphocytes are more heavily implicated in the adaptive immune response. So we'll be talking about them in a lot more detail next time. But for today, we're probably going to spend more time talking about those from the myeloid stem cell lineage. This is a nice diagram just to show that differentiation step here. We have our hematopoietic stem cells that arise from the bone marrow. We have the myeloid progenitors and the lymphoid progenitors, and then all of the cells that arise from those two different pathways, which make up most of the complement of our immune system. Many of the cells in the myeloid lineage become what are called granulocytes. Granules are little pockets of death that are found within these cells and can get released upon foreign pathogens to kill them. Or when cells are internalized during phagocytosis, they can be applied to cells internally to kill them that way. Basophils, eosinophils, and neutrophils are all granulocytes, and they'll all play their own specific roles in the immune system, but the granules are usually at the center of their mechanism. Not only do these granules act to kill bacterial cells or viruses, but they can also stimulate other signals for our immune system to indicate that there's an infection present. And it's almost like pulling the fire alarm in a way. It'll communicate to the body that there's an issue in a specific area that requires attention, and it'll actually cause other cells to show up at that site to do even more work. So it's like calling for backup, putting up the bat signal, if you will. Any way that you'd like to think about it, a lot of these granules can tip off and amplify our immune response. Monocytes are an important kind of cell because they are always circulating in our blood looking for trouble. And when they do encounter it, they can undergo a differentiation into one of the pivotal parts of our immune system called the macrophage. Macrophages are enormous cells that actually kill on sight. So they show up at a site of infection, they engulf the bacteria that are considered to be foreign, and they just start destroying them. They do one cell after the other until the infection is cleared. They do this alongside a variety of other cells that are doing their own type of mechanisms, but macrophages are very integral to getting our immune system off the ground. And this here is a wonderful photograph showing a macrophage. So you can see how large that it is compared to these cocci here that are all huddled together waiting for death to get them. But you can see that through the phagocytic process, where the cell will extend its cell membrane, engulf all of these cells, take them inside in the endocytic pathway to the lysosome, they'll get degraded and killed. And once these cells are dead, obviously they can't keep dividing, but macrophages are also an important link to our adaptive immune response. But we won't touch on that just yet. Dendritic cells are the other big phagocytic cells that are found at these common portals of entry in large numbers. And they do similar work to macrophages, except that they don't kill on sight. Dendritic cells are more like messengers, where they'll be fighting alongside macrophages, but they're going to do a lot of that liaison work. They will be engulfing pathogens and then carrying them away to other sites, where other immune cells are just hanging out, waiting for the next alarm to go off so that they can launch themselves into action. And dendritic cells play a very large role in taking that message to them and getting them to become activated so that they can go and start to actively defend ourselves against whatever infection has. Antigen presentation is what that's called, the messenger aspect, and we're going to talk about that more when we get into the adaptive immune response. Now, contrary to popular belief, despite having the name dendrite, like we've probably heard of when we talk about different nerve cells, they're not related. They do, however, have a similar physical characteristic with these long little arms or appendages or dendrites that project off of them. This actually helps immunologically speaking because they can interact with a lot of different cells at once. Anytime you increase that surface area, think of things like villi in the gut that help to increase surface area for absorption of nutrients, right? It's the same principle that's applied here. The more of these little appendages that extend, the more ways that these cells can touch more things at once, instead of them just being one little round blob. The lymphocytes are probably the stars of the immune response because of how heavily they are involved in the process, and also because they're going to be the ones taking the most action. They're either the delegators at the top or the ones that are on the ground, really ramping up the immune response as needed, but we're going to talk about them more next lecture. So for our purposes for this lecture, I guess this is a great summary diagram to concentrate on, looking at the different cell types and then whether or not they're granulocytes, whether or not they're phagocytic. That would be a good place to really concentrate on for some multiple choice questions that will ask you to differentiate the different types from one another. We'll spend some time now on phagocytosis, which is a type of endocytosis. And this is used when particles that are very large need to be taken up into the cell. We've talked about a couple of different types of phagocytic cells, but primarily macrophages, dendritic cells, and also neutrophils are the most relevant ones for our immune response. Phagocytosis is used in two major mechanisms, where we either have the engulfment and destruction of something that is antigenic, or you have the engulfment, destruction, and presentation of that to another cell to amplify that immune response in those secondary lymphoid organs away from that initial site of infection. This is a great diagram here showing the microbe or another particle, again, it could be a non-living thing, that gets detected by a cell as foreign. The cell is going to extend its membranes around the entity, and this is done all through the cytoskeletal rearrangement. You have actin that's going to be building at those edges, helping the membrane to extend and be stabilized as it reaches around whatever it's going to engulf. And then that entity gets brought into the cell, and then you're going to see a fusion with the lysosome. The lysosome, if you've forgotten, is an enzyme-packed acidic little pocket that when fused will help to digest and destroy the entity that's inside, and then those little parts inside are dead. Now, depending on the type of cell, that can just be secreted out into the extracellular environment like we see here, or some of these little bits and pieces can be mounted onto a protein and just stuck outside the membrane as a little signal, and then that's what gets presented to other cells. So if you're another immune cell, and you see that little signal kind of sticking out of the cell membrane, then you can become activated and go and fight the good fight. This is a very cool picture showing some diplobacilli here that are being engulfed by an immune cell. So you can see again that extension of that cell membrane as it's reaching around these two cells to engulf and probably destroy them. We've been talking a lot about bacteria, but some pathogens, as we've discussed, are large. They can be worms, they can be parasites, they can be multicellular, and in these cases, we have to get something in place to destroy those things as well. Phagocytosis won't cut it in this case, so macrophages and other kinds of immune cells will actually start to secrete a lot of degradative enzymes to slowly break these things apart piece by piece. As they attack the invaders and chop them down into smaller bits, those pieces can be phagocytosed and dealt with. It sounds pretty gruesome, but it is required. Cytolysis is another mechanism through which some of these immune responses can function, and this is again causing that breach in cellular membranes to cause them to erupt, which is another very effective way to kill cells. In the busy world of our bodies, you might be wondering how our immune system can actually tell when something is foreign or antigenic, because there's a lot of work to be done. It's not as if cells have eyes that can go around and pick out different things that look a little strange. There's a lot of time and effort and energy that's put into this monitoring. So sometimes we can implement different mechanisms to help flag things a little bit better. One of the ways that we do this is with the complement system. There are about 20 different kinds of proteins that exist in this small mechanism that can be applied to and bind to cells that are pathogenic in nature. This can signal to our own immune system that these cells are foreign versus waiting for our immune cells to do the work to discover that on their own. At the center of this whole mechanism, and it's quite large and complex, but we won't get into it in too much detail here, but there's one protein here called C3, and when it gets cleaved during the complement mechanism, we end up with a part of it that will become an opsonin and another part that will induce inflammation. C3b as an opsonin is that flag that we've been talking about. Opsonization is simply the coding of something, so it's like sticking a flag onto the back of something to identify it. And these opsonin proteins will bind and stick to the surfaces of pathogens, and when our immune cells see them, then they know that that's something that needs to be attacked and destroyed. The inflammatory part of this simply makes it so that other cells are drawn to the area where these pathogens are present, so that the signal actually gets seen. So one of these is like shining a spotlight on an organism. The other one is going over the loudspeaker and saying, "Hello, is anyone in the area? Please come by because there are some pathogens here that need to be taken care of." So this diagram shows this in a little bit more detail here. Opsonization, again, you can see bound protein to the surfaces of these bacilli, which will result in them being phagocytosed more easily. We have the inflammatory response that gets induced, where we're going to have a signal being sent out to our immune cells in the bloodstream that can then go through extravasation and exit that into the surrounding tissue towards the infection to help fight the fight. And then thirdly, there's cytolysis, where our complement proteins, in some cases, can actually form a pore in bacterial cell envelopes and cause them to die as a result of that. So the complement system is quite complex and is one of the major constituents of our innate immune response. We can get into a ton more detail here, but we'll leave it there. All that you really need to know is that it does these 3 different functions and does them well to help us to survive and continue to persist on Earth. This diagram just takes these concepts a little bit further, and of course, this is more just for your interest versus something I'd like you to spend time memorizing or focusing on, but these are actually the 3 pathways that make up the complement system as a whole. The most important takeaway here is that all of them converge at that C3 enzyme that we talked about before that gets split and can go off to do its different roles. But at the end of the day, the other important part is the removal of immune complexes, opsonization of pathogens. Both of these are code for destruction of the pathogen that is infecting a host. Some bacterial species are actually very effective at evading our phagocytic mechanisms. We're going to have an entire lecture dedicated to pathogen evasion strategies where they will run into our immune cells and then talking about how they actually get away from all of the efforts that our immune system makes to kill them. But these are just a couple of examples. Some of them we've already discussed here, like how staphylococci will produce things like leukocidins that will just plain destroy phagocytes. So if they don't exist, they can't be engulfed and destroyed. There are some other types of bacteria like listeria that will get into the phagosome, so that endocytic vesicle that they'll come in with. And then they'll just bust out of there and live in the cytoplasm of the cell and can actually travel from cell to cell without ever leaving back into the extracellular environment again. A very tricky disease state to treat. Salmonella species can inhibit the fusion of the phagosome and the lysosome, so they just live inside of that endocytic vesicle afterward. And obviously we talked about capsules and how they can prevent phagocytosis because it prevents the immune system from even being able to recognize those antigenic components to begin with. Fever is also something that we use quite a lot to combat bacteria, and it's defined as an abnormally high body temperature. Pyrogens are fever-causing agents that get detected on foreign entities. We talked about this a little bit previously. Things like lipopolysaccharide can actually induce the fever response just by being an endotoxin present in the body. As a result of detecting that signal, our bodies are actually going to go through a number of physiological changes because of the fever that gets induced. Blood vessels are going to constrict to prevent heat loss. We're going to increase our metabolic rate. And then we are also going to start to shiver, which is going to increase our muscular activity to create even more heat. A combination of these things results in fever. Our high body temperatures are going to render bacterial enzymes in a state where they're unable to function. This is done usually through denaturation. There's that very small threshold that if we push proteins too far past it, all of that tertiary structure is lost. And when we consider how important the enzyme active site is, when we look at its efficacy of binding with its substrate, when any of that is even mildly compromised, the efficiency of that enzyme is going to decrease tremendously. Another thing that can happen during denaturation is the exposure of those hydrophobic components that are normally internalized in proteins. And when all of those internal components just start showing externally, that means that many proteins are going to be attracted to one another because hydrophobic molecules are attracted to other kinds of hydrophobic molecules. So not only are these proteins unfolding, but then they're going to get stuck together in a way that's going to prevent them from being able to ever be pulled apart again. That's called aggregation. So denaturation not only impacts that physical structure of the enzyme itself, compromises that tertiary structure, but also makes them irreversibly bound to other proteins. Now, in humans, we have the ability to prevent denaturation from happening altogether. This is done through our heat shock mechanisms. And heat shock proteins are called chaperones. So as proteins are beginning to unfold, heat shock proteins come in and start to bind onto those hydrophobic regions to prevent them from being bound by other proteins that are also unfolding. It kind of holds them together like glue. But when we look at bacteria, they don't have those mechanisms. And as a result of that, when they denature, they're denatured. They can never refold their proteins after that and usually die as a result. The inflammatory response in general is characterized by redness, pain, heat, or swelling at the site of infection or injury. And this is caused mostly by the cell response by the host. As our blood cells exit en masse into sites where there are things a-brewing, we're going to have obviously more fluid building up there, causing heat as well, and also pain because we're distending those tissues as pooling is occurring. Inflammation is important because it will help to destroy the injurious agent because we're getting a lot of help to those sites to help to fight that fight. But it'll also limit the effects of the agent by isolating it. One big part of that inflammatory response is blood clot formation to help to wall off that infection from being able to spread outward. And by taking these and many other steps, we can eradicate a lot of things from our bodies before they have a chance to take hold. Inflammation is normally going to be mediated by endothelial cells. Epithelial cells line our common portals of entry in most of our tracts. Endothelial cells, on the other hand, line our blood vessels. So they are the innermost layer of cells that exist in our blood vessels. In normal resting vessels where there is no threat to the host, the endothelial cell layer is intact, which makes sense, right? You think about our closed cardiovascular system. We don't normally have blood leaking out into other parts of the body where we don't want it to go. But when there is a threat there, that's when things start to change. The threats are usually received in the form of a chemical signal. So if cells are injured or they're being destroyed by an infectious agent, they'll start to release different proteins and other things that can go to the endothelium and say, oh no, there's something happening over here, please help. So what the endothelial wall will actually do is permeabilize, and this will allow for immune cells to detect that signal as well, and then squeeze out of our blood vessels into whatever area of the body that the signal has come from. This is chemotaxis. Chemotaxis meaning movement towards a chemical. So once those cells get out of the bloodstream and get to that area of infection, they can start to execute all of the methods that we were just talking about earlier. Following the phagocytosis, the destruction, the migration to secondary immune tissues to do antigen presentation, tissue repair processes can begin. And the immune system is very heavily involved in that as well. Many of our immune cells that work to fight off these infectious agents also work to help to rebuild the tissues after the fight is finished. So here we have a nice diagram where we have some bacteria entering on a knife. I always find this so amusing that they chose this in particular, but you can see here we've penetrated through the epidermis into the dermal layer, and we have bacteria that are introduced near our bloodstream. After a while, we have a lot of chemicals that are being released by our skin cells to say, ow, we're injured, please send some help over, there are infectious agents here. And then you can see that as a result of this, there's increased blood flow to this site. There is pooling of immune cells that are shown in blue here that are moving out of our vessels into that tissue, and they're going to start to engulf and destroy all of those different organisms over time. You can see some more of that here. The extravasation is these cells squeezing through the cells of the endothelial layer into that interstitial space where the infection is present. And then after everything is destroyed and taken care of, you can have the healing process that begins. We've mentioned cytokines once in a while throughout the course here, and these are regulatory proteins that pretty much allow cells to communicate with one another. There are a slew of them, tons and tons. We could spend an entire course just talking about them and their own functionality, but they control pretty much everything that cells do. In the context of the immune response, though, we always class them as either pro or anti-inflammatory as regulatory mechanisms to help us to control the immune response and when and where it's going to occur. Inflammation, as we've discussed a couple of times, is important for the immune system to do its work, but when it's chronic or unwarranted, it can be very damaging to the host. So having this mechanism highly regulated is extremely important for host health. So this is a table here just outlining a handful of the many, many cytokines that exist. Once again, nothing that I want you to spend time memorizing, but just as a little bit of a snapshot of some of the functions that these things control. Everything from cell growth to cell death, to differentiation, to migration, communication, the list goes on and on and on. We've talked about interferons a little bit in the past, but we'll bring them back here now in the context of the immune system. This is a specific type of cytokine that can be either antiviral or antibacterial in nature, but more or less whenever you have a pathogen that gets bound by a host cell, our cells have an opportunity to alert other neighboring cells that there's a problem. What this does is allows for other host cells to prepare to be able to defend themselves against an infection before it's too late. So more or less, if one cell gets infected by a virus here, the cell that's infected is going to say, oh no, it's too late for me, but I'm going to try to send out something to alert my neighbors. So it's actually going to have genes in the genome that can be transcribed and translated into a signal. Again, cells will signal with proteins. They don't have voices, obviously. So this signal will get produced en masse, released from the cell, and then its neighbors here can get that signal. So once they are aware that this virus is in the neighborhood, then they can start to ramp up their own defenses. They start to produce antiviral proteins that will protect them from the same infection. And here now this virus cannot penetrate this cell and continue to do its nefarious work. Another class of cells that's really important to us are natural killer cells. And these are really great at looking for cells that are infected or cancer cells. I've mentioned a couple of times that even though our external chemotherapeutic interventions are not usually very good at getting rid of different types of microorganisms that are intracellular in nature, this leg of the immune response with natural killer cells as well as cytotoxic T cells, which we'll talk about later on, are integral for identifying and destroying cells that are infected. So they can tell them apart from normal cells that are healthy and know which ones to destroy and which ones to leave alone. So hopefully this was a good introduction to our immune response. We're going to continue next lecture with some discussions around the adaptive immune system. And just so that you know, I also teach a course in third year called Immunology. So if this is something that interests you, that course does get offered every once in a while. So maybe consider taking that in the future. I will leave it here for now. And next time we'll be talking about some adaptive immunity. If you have any questions, please let me know. You can email me at laurar@nipissingU.ca. And thanks so much for now. Bye.